for Science of Synthesis at Thieme Chemistry. And today we have Shu Kobayashi chairing our session with speakers Emma Davison, Chun Hei, Tomohiro Hattori, Hyunwoo Kim, Xiaoyu Yang, and Durga Prasad Hari. So before we start today, I will just do a couple of housekeeping points. Today's seminar is being recorded. That's so that others who haven't been able to make the live event will be able to watch the session afterwards. And to avoid interruptions during the presentations themselves, we've put the audience on mute, and you'll be kept on mute throughout. There's no live Q&A today, but we would encourage you to put any questions that you have into the chat function of Zoom, and we'll come back to those and answer as many as we can after the session is finished. You can also tag us on social media using the Thieme Chemistry link on the screen right now. And if you'd like to hear more about our Cheminar series or subscribe to our Thieme Chemistry newsletter, you can do that using the links here as well. Before I hand over officially to Shu, I'd like to talk a little bit more about Thieme Chemistry itself, the portfolio. For those of you who aren't aware, Thieme is a German company with an international presence and over 130 years experience in publishing science and medical information. And Thieme Chemistry is a branch within the Thieme Group. We've traditionally specialized in organic synthesis, so helping chemists acquire knowledge about organic chemistry, as well as helping to devise efficient synthetic strategies. You may well be familiar with our journals Synthesis, Synlet, and Synopen, as well as Sustainability and Circularity Now, which has broadened our scope to involve areas such as sustainability and green chemistry. We also have a suite of biomedical journals for which there's a similar bioscience series on Cassini. Today's Cheminar is presented by Science of Synthesis, which is our text and structure searchable online reference work and covers methods in synthetic organic chemistry. And these critical reviews are written by some of the top people in each area and aim to provide a definitive overview of the most important methods for organic chemistry. Science of Synthesis covers a broad range of chemistry, for example, hot topics such as base metal catalysis, DNA encoded libraries, etc., and more traditional chemistry, for example, the synthesis of thioamides or furans. Our chair today is Shu Kobayashi, and he's professor of organic chemistry at the Faculty of Science at the University of Tokyo, and he's received numerous awards, among them the Nagoya Silver Medal, the Negishi Award, and the Japanese Medal of Honor with Purple Ribbon. Shu's long been associated with Thieme Chemistry and joined the Science of Synthesis editorial board in 2017, having also edited the Science of Synthesis volume on water in organic synthesis. The editorial board comprises renowned chemists from both academia and industry who select the topics covered, and the volume editors that edit the series. The Science of Synthesis Early Career Advisory Board are up-and-coming rising stars in chemistry and are selected by the editorial board of Science of Synthesis. So it's 30 people from all over the world, 6 of whom we have with us today, and they have a range of research interests also in areas adjacent to organic synthesis, and they provide us with advice and inspiration, making sure that we have a global perspective of organic synthesis today and where it's going in the future. All of today's speakers are also past recipients of the Thieme Chemistry Journal Awards, and these are made by the editorial boards of the Thieme Chemistry journals, and they recognize upcoming chemists at the beginning of their careers, and they're awarded each year to an international mix of people too. So now I'd like to hand over to Shu so that he can introduce the first of our speakers today. Shu, the floor is yours.
Okay, thank you, Karen, for the very nice talk. And I'm Shu Kobayashi at the University of Tokyo. It's my great pleasure to start Thieme WebCheminar today. And there are, as Karen mentioned, there are 6 speakers, mainly from the Asian area and Oceania. The first speaker today is Dr. Emma Davison. Emma took PhD at the University of Auckland in 2018, and then after several postdoc experiences, in 2023, Emma co-founded Cloudburst Biotech, and last year, 2024, she joined the Auckland University of Technology School of Science as lecturer in chemistry. Today, he's going to talk about practical and concise synthesis of nucleotide analogs. Emma, please start when you are ready.
Thank you, Professor Kobayashi, for the kind introduction. I'm just going to send a request to share my screen. Oh,
Okay,
are you
that's
able
fine.
to see my slides okay?
Okay.
Great. Thank you, Professor Kobayashi, for the kind introduction. Today, I'm actually going to be discussing some of the research that was conducted during my postdoctoral position at Simon Fraser University, exploring the de novo approaches to nucleoside synthesis. I presently lead a research group at Auckland University of Technology, and while I'm unfortunately unable to discuss this independent research, the work that I am about to discuss today did inspire my present research directions. So without further ado, nucleoside analogs remain one of the most important classes of small molecule drugs as both antiviral agents and anti-cancer agents, and they're increasingly important building blocks in the development of RNA therapies, and that includes both mRNA vaccines and oligonucleotide drugs. Modifications to both the sugar unit and the nuclear base are both essential for controlling drug pharmacokinetics and pharmacodynamics. Traditionally, though, accessing these modified nucleosides has been synthetically demanding. Most synthetic routes still rely on the Volbruggen glycosylation, which occurs between an acylated ribose sugar and a silylated nucleobase. Mechanistically, this reaction proceeds via formation of an oxocarbenium ion, and that's followed by neighboring group participation and the formation of a cyclic cation, which can then be attacked at the anomeric position by the most nucleophilic position of nitrogen of the silylated nucleobase. While this chemistry is very robust, and it has been around for a long time, the approach comes with a lot of well-known drawbacks. So it clearly relies on chiral pool ribose starting materials, and these require a significant number of protecting group manipulations if one is wanting to access chemically modified ribose substrates, which can add to a lot of steps to the syntheses of these substrates. And additionally, the glycosylation step also produces mixtures of regioisomers, which can give really poor distereoselectivity and regioselectivity as well with modified ribose substrates. A significant advancement was made in 2014 when David McMillan's group disclosed a de novo synthesis of 2' modified ribose rings using a Mukayama-Aldol reaction to set that 2' stereochemistry. This reaction was followed by a reductive cleavage of the TMP group and an acid-mediated cyclization to afford a 2' modified ribose sugar, which is quite efficient and stereoselective synthesis. However, this ribose first approach still relied on subsequent installation of the nuclear base using that Volbruggen chemistry, and so some of those limitations of that classical reaction still remained. And this was until 2020 when Dr. Robert Britton's research team, led by Mike Meanwell, pictured here, introduced a ribose last approach, which essentially completely avoids the use of this Volbruggen chemistry and its inherent selectivity and protecting group challenges. This methodology uses two simple achiral building blocks to construct the ribose framework around the nuclear base in the final step of the sequence. It's a scalable process and enables access to a diverse array of modified nucleosides, as you'll see in the rest of this talk. And these can be accessed in only 3 to 4 synthetic steps. So looking a little bit more closely at this reaction sequence, the first step is an alpha-fluoroaldol reaction, which uses NFSI as an electrophilic fluorine source. This step produces a mixture of fluorohydrin epimers, which are inconsequential for the final cyclization step, as you'll see. These epimers are formed through slow epimerization of the fluorinated aldehydes. And importantly, the use of L-proline in this reaction is able to catalyze both the fluorination step as well as the enolization of the dioxanone substrate, which will form an enamine intermediate pictured here that can then undergo an aldol reaction and the stereochemistry by promoting refacial attack of that aldehyde. The mixture of these fluorohydrin substrates can then be reduced with tetramethyl ammonium triacetoxyborohydride. This reagent is essential for delivering the syn-diol product selectively. Alternatively, a Grignard reagent can be used to introduce C4' functionality, which I'll come back to later. The final step is an annulative fluoride displacement, an AFD reaction that occurs through an SN2 type mechanism. In the case of the syn-diol, this can cyclize directly to give the beta anomer, which is the naturally configured nucleoside, or alternatively, the anti-fluorohydrin can cyclize to the alpha anomer, which then under these basic conditions will epimerize in a one-pot procedure to give the more thermodynamically stable natural beta anomer. The nucleoside formed through these reactions is typically formed with high enantioselectivity. selectivity. Most nucleosides are formed in greater than 95 to 5 ER. And so while this chemistry is exceptionally powerful and scalability was demonstrated on several examples, a lot of the procedures reported were on a much smaller scale. And so the next challenge within the group was to create a generally scalable and broadly applicable process that would enable more widespread and reproducible use of this chemistry across both academia and industry, who would both find a lot of value from this work. So as such, I developed a comprehensive step-by-step guide for this methodology. The guide was developed to include troubleshooting tips for common issues and highlighted potential hazards. It also provided alternative procedures for problematic substrates. Our aim was to make it really straightforward so that even new graduate students could follow and reproduce the chemistry reliably. We chose to optimize the protocol on uridine for a few reasons, but primarily because it presents a particular challenge, actually, due to its high polarity and solvent solubility issues. We were able to achieve this robust research scale reaction of uridine, the acetonide protected uridine on 2.8 gram scale, and using just typical equipment that's found in academic labs. And we collaborated with Merck's process research and development team, led by Elsie Campo and Steve Silverman, pictured here, who translated the procedure through this collaboration to a larger process scale of 30 grams using equipment typically found in industry labs. And this work was published in Nature Protocols in 2022. So just looking at these steps a little bit more closely in terms of the protocol. So the first alpha fluoroaldol reaction, the heteroaryl aldehyde can actually be used either as the hydrate form, the aldehyde form, or a mixture of the two. And this doesn't have any consequence in the reduction. The addition of the N-fluorobenzene sulfonamide, the NFSI, is an exothermic reaction, so a careful temperature control is essential. On the research scale, we perform this reaction in a fridge with stirring to ensure reproducibility and safety were both met. But when scaling up this process to industry scales, the reaction was conducted in a cold room to manage that heat evolution effectively. Isolation of the resulting fluorohydrin product from the DMF solvent actually presented quite a challenge, especially on these larger process scales. And so to address this, we recommended employing short path distillation to remove the residual DMF prior to column chromatography. The next step was the reduction of the fluorohydrin to give the syn-diol. On the research scales, this was conducted at -15 degrees Celsius in a freezer. Interestingly, we actually found that continuous stirring was not required at the scale, and we obtained similar results whether the reaction mixture was stirred continuously or actually left static. On process scale, the reaction was carried out in a jacketed 3-liter vessel connected to a chiller unit, which maintained those cold temperatures, and overhead stirring was required on the scale to ensure uniform cooling. The workup on the process scale was adapted to use of equipment typically found in industry labs as well, of course. The final step of this reaction is the annulative fluoride displacement. This is operationally simple as a reaction setup, just stirring at room temperature with aqueous sodium hydroxide and acetonitrile. Most substrates are obtained in sufficient yield just with a standard aqueous workup protocol. However, for really polar nucleosides like the acetonide protected uridine here, an alternative procedure was developed to help improve the recovery of these products. And this involved neutralization of the reaction mixture, followed by a lyophilization step, and actually just dry loading of that crude solid material straight onto the column for purification. While that second workup B, was a bit more labor intensive, required access to a lyophilizer and obviously long wait times during that freeze drying process, it did give significantly better recovery of the polar products like the acetonide uridine here. On a process scale, lyophilization is of course not practical, and so the workup had to be adapted to a more scalable approach. This actually involved a more extensive extraction procedure during the workup using DCM and isopropanol mixtures to recover the polar product. However, that is not required for all substrates. Less polar products do not have such issues. So the parent protocol that we developed can be adapted quite simply to access other analogs. One of the strengths of this protocol is that the final nucleoside is inherently protected at the 3' and 5' alcohols as an acetonide. And so this inbuilt protection enables subsequent selective functionalization at the 2' position without additional protecting group manipulations. And this was demonstrated by synthesizing a key 2' modified precursor to the antiviral drug aprifosbuvir. And this was obtained in just 5 steps from these simple achiral starting materials. Alternatively, the parent protocol can be adapted to access 4' modified nucleosides. So in this approach, in the original papers, the fluorohydrin intermediate undergoes a Grignard addition to install the 4' substituent. And if the crude Grignard addict is simply warmed to room temperature, then the magnesium alkoxide species can undergo the AFD cyclization step directly, giving an alpha L configured nucleoside analog. Alternatively, if the reaction is quenched and then treated with Lewis acid, such as indium trichloride or scandium triflate, then this can allow isolation of either the alpha or beta D configured nucleosides. The stereoselectivity of this Grignard reaction under these original conditions was largely substrate dependent and produced mixtures of anomers and unnaturally configured L sugars. This is, of course, useful for small-scale medicinal chemistry work where unnatural sugar configurations might be desired. But of course, stereoselective synthesis of the natural beta-D configured nucleosides was a priority for subsequent studies. These studies were led by PhD student Garrett Muir, and they have recently been published this year in Nature Communications. Essentially, these studies revealed that a non-coordinating DCM solvent and use of specifically the organomagnesium iodide species gave the best diastereoselectivity for formation of the syndiol and therefore the naturally configured beta-D nucleoside analog. So I encourage anyone listening to have a look at this article if they're interested in learning more about the computational and reactivity studies that led to this really interesting halide effect. Finally, bridge nucleic acids, or BNAs, are really valuable building blocks in oligonucleotide therapeutics, and these can also be accessed using this methodology. So by reacting the fluorohydrin intermediate with an ambiphilic reagent, a C4' electrophilic carbon can be introduced that will undergo a cyclization cascade to form both the ribose ring and that bridging ring in a one-step process. To illustrate this, a thymine BNA was synthesized in 4 steps only using this methodology, which was a really dramatic improvement compared to the 23-step synthesis of the analogous uracil BNA that was previously reported. And this was done on a very large scale, 54 grams. This strategy has been developed quite considerably by Dr. Cohen Huxley, who expanded the scope and enabled the synthesis of a wide range of BNAs using this methodology. And this work was recently published in Angewandte Chemie. For any oligonucleotide chemists in the audience, this methodology enabled access to quite a wide range of different BNAs, including LNA, ENA, C-ethyl-LNA, and spirocyclopropyl BNA in just 3 to 6 steps. Those analogs, some of them are pictured up here in this final scope slide, along with some other specific examples of the methods that I've just discussed. But alongside these, a select number of other examples are shown here. So the protocol has enabled us to access as well nucleosides with different nuclear bases, as well as combination of nuclear base and C2' and 4' modifications. Interestingly, the enantiomeric L nucleosides can also be accessed using this methodology just by using D-proline instead of L-proline in the first step. And finally, the amino or azonucleosides can also be accessed using this chemistry as well. Just to sum up, we really believe that the development of this protocol makes the process of this chemical process accessible to both academic and industrial settings, and enables the reproducible and scalable synthesis of quite a diverse nucleoside analog scope, which we hope will facilitate the translation of these nucleoside analog discoveries to more practical settings in a medicinal chemistry place. So I've already acknowledged most of the individuals and funders who have contributed to this research, but I would like to recognize quickly the Michael Smith Health Research BC, who provided funding for my postdoctoral position at Simon Fraser University, where I contributed to this work. I'd also like to thank briefly AUT, the students presently working with me, and funders, especially Health Research Council in New Zealand, who are supporting me to build my research program in the very early stages at AUT in the field of nucleoside and oligonucleotide therapeutics. And finally, of course, I'd like to thank Thieme for the invitation to present today, and to everyone tuning in for their interest. I'd be happy to answer any questions either in the comments section, or you can feel free to reach out to me on LinkedIn as well, and I can try and respond as quickly as possible. Thank you.
Okay, thank you very much, Emma, for very interesting talk from New Zealand. Okay, let's move to the next speaker. Next speaker is Dr. Chang Hee from Southern University of Science and Technology. Zhang took a bachelor and also PhD in Wuhan University under supervision of Professor Iwan Lei. In 2013, he joined the Mashu Gold Group in University of Cambridge as postdoctoral fellow. In 2018, he started his independent research career at SUSTEC. Today, he's going to talk about new adventures in silicon sterogenic silane chemistry. John, please start when you're ready.
Okay, so thanks, Professor Kobayashi, for your kind introduction. So can you hear me?
Yes, please share your
Okay,
slide.
let me share my slides. Okay.
Okay, that's fine.
Okay. Can you see my slides? Perfect.
Perfect. Please start.
Okay, so thanks, Professor Kobayashi, for your kind introduction, and thank the Thieme Group for organizing these seminars. And it is my great honor to share my research results here. And okay, my group mainly focuses on chiral organosilicon and chiral organoboron chemistry. And today, I'm gonna talk about the story of silicon sterogenic silanes. Right, and chirality is important when I started my independent research, I was thinking about pushing the boundary of chiral molecules. And what fascinates me is silicon. Lying directly below carbon, silicon certainly can also be a stereogenic center. However, organic silicon was omitted in biological evolution. Therefore, chiral signings are not found in nature. So compared to their carbon counterparts, the unique property of silicon, such as the larger atomic radius, longer carbon-silicon bond, and lower electron activities, make us believe silicon stereogenic signings may have the potential to open up a new realm of chiral molecules. So talking about organosilicon, these are very important molecules with valuable applications as functional materials, bioactive molecules, and synthetic tools. However, despite enormous organosilicon compounds have been developed, silicon stereogenic signings in initial enriched form have been much less explored. The history of silicon stereochemistry starts from 1907, really long time ago. However, progress in these fields has been remarkably slow. And for a long time, the synthesis of this silicon stereogenic signin was considered one of the most challenging tasks in synthetic chemistry. Why is that? One of the major reasons is the silicon double bonds is not as stable as carbon double bonds because of the poor p-pi orbital overlap. Therefore, the addition reaction for the generation of the chiral silicon center is not feasible. Instead, the typical approach relies on the desymmetrization of the per-chiral sp silicon atoms. However, the larger atomic radius and longer carbon-silicon bonds usually prevent the formation of a compact transition state, making any discrimination of the anisotropic groups on silicon difficult. So in the last decades, a number of transition metal-catalyzed desymmetrization reaction has been developed for the synthesis of these silicon stereogenic signins. Although with some success, most of these reactions suffer from low EE, poor functional group tolerance, and limited scope. So there is an urgent need for new catalytic strategies towards silicon stereogenic signins with high efficiency, high EE, and diversity. So we questioned ourselves, can we develop a general and efficient methods for the asymmetric synthesis of silicon stereogenic monohydrosignins? We focus on monohydrosignins because normally they are bench stable, while under certain conditions, you can transfer the chiral silicon hydrogen bond stereospecifically, making them a perfect plan for molecules. So the idea is we use the per-chiral dihydrosignin as the starting materials, which was activated by chiral transition metal catalyst for the generation of this key silane metal species. Then this silane metal species react with CH or XH coupling partners, give these target chiral monohydrosignins, and we call it silicon CADC reaction. From a synthetic point of view, the starting materials, dihydrosignins, are readily available, and this reaction is dehydrogenative couplings. Presumably, dihydrogen is the only byproduct. And more importantly, we are running coupling reactions. That means we can use different coupling partners, like CH bond, OH bond, or NH bond, which allow us to synthesize diverse chiral signin product. So the strategy looks attractive. However, the challenge is how to precisely control the reactivity, chemoselectivity, and the initial selectivity, because there are many opportunities the reaction can go wrong, leading to the side product. For example, the homocoupling, redistribution, and overreactions. So dating back to 1970s, Korean and co-workers made efforts to develop these intermolecular silicon-oxygen dehydrogenative coupling reactions. Unfortunately, at that time, only 90% EE was obtained. In 2003, Leighton developed a copper catalyzed reaction. He got 9 to 1 DR, which is not bad. However, in this reaction, this particular initial enriched chiral alcohol has to be used as a substrate. And then in 2013, Taka and co-workers developed an interesting intramolecular silicon carbon dehydrogenated coupling reactions. By using these special bis biarene dihydrosianes, they realized intramolecular double CH silylations for the synthesis of this spiral silane bifluorine product featuring actual chiralities. They got decent results. However, in their later mechanistic studies, they found out the reaction is quite messy in the early stage. And the good news is that all these intermediates in the end will turn to the final product, so it's fine. However, if you want to isolate one of these monohydrosianes, that would be very challenging. So, standing on these pioneers' shoulders, we started our research by using these dihydrosianes to conduct an intramolecular CADC reaction for the synthesis of these cyclic monohydrosianes. Long story short, we are very lucky to find a rhodium-Josephus catalytic system which allows us to get this product. And the rigid, bulky Josephus ligand effectively controls the interaction force via its asymmetry. And key to the success is the precise control of the silane rhodium metal species in the process. So here is the catalytic cycle. In general, the reaction starts from the rhodium-1 hydride species A, which undergoes oxidative addition with one of the silicon hydrogen bonds, gave the intermediate B. Then reduction eliminations with the release of hydrogen gave intermediate C. And from here, the next step is CH activations form the intermediate D. Next, after the carbon-silicon bond reduction eliminations, presumably you will got the final product and regenerate the rhodium hydride species, right? So it's quite straightforward. However, in our later mechanistic studies, we found a very interesting resting state in this catalytic cycle by the phosphine NMR. So to figure out what is the structure of these resting states, then we carry out the stoichiometric reactions. So treatment of this rhodium-Josephus chloride with the substrate dihydrosianine in 1:1 ratio at room temperature, we got a really beautiful yellow solid. And its phosphine NMR is exactly the same as the resting state in the catalytic reaction. It's double DD signals. And for the proton NMR, it's also very clean. We can assign all the organic parts. And you need to be careful. We found out at the -6 to -7, there is a broad and weak peak. And then we realized it could be the metal hydride. Then we carry out the lower temperature NMR studies. At -40, we got a high resolution of the rhodium hydride species. And it looks like it has 2 hydrides. And in the end, we are lucky to grow the crystal structures as shown here. Indeed, this resting state, the carbon-silicon bond has already formed, and both hydrides go to rhodium. So let's put this resting state back to the catalytic cycle. You will immediately figure out what's going on. So as I said, since here, the intermediate D, after carbon-silicon bond reduction eliminations, it will generate the product. However, this one still has a silicon hydrogen bond, which would undergo the second oxidative addition, leading to this resting state. And in our later DFT studies, it seems like from intermediate D to intermediate D is more like a continuous silane metal migrations. So with these understandings, to facilitate the release of the product and maintain the high EE, we add alkene to in situ trap this rhodium hydride. So after insertion, we got intermediate F. So here, the carbon-silicon bond reduction eliminations will give us the tetrasubstituted silane product. We can get 99% EE, which is very encouraging. Of course, this is not our target molecule, right? We want the monohydrosianes. So next, when we switch the alkene to a bulky one, like noborins, after insertion, because here is more sterol hindrance, which prefer undergo silicon hydrogen reduction eliminations rather than silicon carbon reduction eliminations, give us this target monohydrosianes with 92% EE. So with these insights, we are pretty much unlocking a general platforms towards these cyclic silicon sterogenic monohydrosianes we can synthesize like 5 membrane, 6 membrane, 7 membranes, a very good functional group tolerance. And for some substrate, we can construct both planar chirality and axial chirality. So with this initial enriched monohydrosilane in hand, you can prefer any stereospecific transformation that you want. For example, you can do the hydrosilylation with alkene, alkyne, carbonyl groups, or coupling reaction with alkynides without the loss of EE. And for this one, you can oxidize it to C-nano, or under the Thieme condition to cut out this carbon-silicon bond, you got the diol, and the actual chirality is still there. So next, we want to push forward of this CADC reaction to the more interesting intramolecular reaction. As you can imagine, the intermolecular version is more challenging because of the loose transition state, more conformational flexibility, and the facile side reactions. So how are we going to address these challenges? So the initial idea is we should find a more effective ligand, right? However, at that time, we try out all those chirophosphine ligand we have. Unfortunately, we cannot get the satisfactory results. So how are we going to do? So with these mechanistic studies, we gradually realize when the hydrosilane react with the metal, it bonded to the metal center. It's more like a X-type covalent ligand. So besides Thieme the chirophosphine ligand, we thought maybe we can Thieme the substitution on silicon, which may enhance the stereo control and inhibit these homocoupling and overreactions. So here is a proof of concept. We use this tributylphenyl dihydrosilane react with simple alcohol at similar conditions. At room temperature, we got really good results, 99% EE, high efficiency. This is really, really interesting. So to figure out why we can get such high efficiency and high initial selectivity, so we conduct DFT calculations with calibrations. So here, due to the time limitation, I just tell you the key steps. The first 2 steps is more or less the same as the intramolecular one. And the next step is the key activation of the OH bond, right? When we calculated this process, follow the literature, the direct oxidative addition or sigma bond metathesis, we found the energy barrier is too high, which is not possible. And then we thought, as we know, this C-like group bonded to the metal and the second hydrogen have some interaction with the metal. And then because we use alcohol and the oxygen atoms have a strong interaction with silicon, we thought it could be a concerted process. So it goes like this. The oxygen attacks the silicon, and here, this is hydride attacks the metal, and the metal gave out the electron to attack the proton. So it is a concerted, more like a proton hydride transfer process. And from here to here, the metal rays is valent. So it is a new type of oxidative addition, which is really interesting, we thought. And the energy barrier is only 11 kcal/mol, which is consistent with our experimental results. And for the stereo control, and this step is the initial determining step. And the S configuration of the transit state is 3.2 kcal/mol lower energy than the R configurations, which explain the 99% EE. So here is what we understand. And for the methodology developed, is much more straightforward. So when we use alcohol to react with dihydrosilane, you got chirosilane ether. You use water, you got chirosinano, and you use heteroarenes, and then you will got chiroarylated silane. We can even use amine, and then we got chirosilanes. So here is the substrate scope. As you can see, we got really good results of the yield and the initial selectivity, and the functional group tolerance is great. Before our work, it's really difficult to get these diverse silicon stereogenic silanes. Okay, and this is one of my favorite reactions. The dihydrosilane react with 1 to 2 equivalent water to get the chirosinanos. We can lower the loading of the catalyst, we still got high efficiency. By using our process, we can easily get this initial enriched chirosinano, and after the second stereospecific hydrosilanations, we can get this bioactive chirosinano product. Back to 1990s, Takaya tried to make this one. As you can see, he needs several steps using metal reagent and only got the racemic one. He needed tedious resolutions to get the initial enriched one. Okay, the last part. Probably you're gonna ask me the questions. Can you avoid using the bulky group on the substrate? Otherwise, it is a limitation, right? So how are we gonna fix the problems? So the answer is still go back to the ligand, because we realize the CELA group play a crucial role in the process. We thought, why not we put the CELA group on the ligand and developed a new type of chiral-CELA ligand. So recently, we developed this chirophosphine CELA ligand. It is a typical LX type ligand, and it can react with the low-valent metal to get this metal complex. For example, if you react with the nickel 0, you got a nickel 2 complex. And very recently, by using these Lico catalytic systems, we developed the second generation of this CIDC reaction. As you can see, for this reaction, we can tolerate the less hindered groups like methyl groups, aliphatic groups, and the functional group tolerance is even better than the rhodium catalytic systems. And follow this procedure, we can make different silicon sterogen silanes. I want to mention these two products. So here it has methyl group, ethyl groups. We can get 90% ER, which is really difficult previously. And for this one, we can tolerate alkyne groups. Previously, we cannot do this. Okay, so here is a summary. And during the last couple of years, we developed a general CIDC reaction for the synthesis of these silicon stereogenic silanes. And currently, we are hoping to explore their functions and applications in materials or drug moleculars. So, finally, I'd like to thank my hardworking students and our collaborators and also the funding support. So, okay, so thank you for your attention. Thank you.
Thank you. Thank you very much, Joan, for very exciting talk.
Okay.
Okay, let's move to the next speaker. Please close your slide. Thank you.
Yeah, thank you.
Okay, next speaker, Dr. Tomohiro Hattori from the Tube University, Japan. Dr. Hattori received his PhD in 2017 under the direction of Professor Hironao Sajiki from GIF Pharmaceutical University. And then he joined Professor Hisashi Yamamoto's group at Tube University as an assistant professor and promoted to associate professor in 2023. Today, Dr. Hattori is talking about peptide chemistry, the title Convergent Peptide Elongation using unprotected amino acids. So, Professor Hattori, please start when you are ready.
Okay, thank you for introduction, Kobayashi sensei. And can you hear me?
Yes.
Okay, thank you. And hi everyone. I'm Tomohiro Hattori in Tube University in Japan, and I'm very glad to chance to talk in this web seminar. And I'm very honorable to share about our research. And this is the first slide and my self-introduction slide, but I have no extra time, so I'll focus on only the very important point. And sorry. And this is thanks to the member of the team that I have been able to join as the member of the Science of Synthesis, ECAB. Then I'm appreciate to the every committee member. Okay, then I'm beginning my presentation. Then I'd like to talk about the peptide formation relating the protecting group. Peptides are the amino acid signature molecule that attractive attention as a representative middle-sized molecule pharmaceuticals. And their sales and their marketing share are increasing year by year. to take advantage of their characteristics. Over 80% of bioactive peptide are prepared by the organic chemical synthesis. Since the new concept of protein group introduced to the peptide bond formation by Bergman in 1932, the cross-condensation between the amino acid with unprotected amino acid have been consistently applied, and the use of protein group is now considered as common sense. But most of these protein groups have large molecular weight and are the cause of large amount of waste. So we wonder whether these protein groups are effectively utilized, and we wonder whether unprotected amino acid could directly use for the peptide bond formation. This is our first question of these topics. Our respective senior of this laboratory have already established the catalytic peptide bond formation using the hydroxy group at N-terminal. So we first apply this system to the N-terminal protein groups. As a result, the use of tantalum, titanium as catalysts, the reaction with the amino acid tertiary butyl ester is proceeding smoothly with the carbonyl group of protein group working as direct group. This direct effect could be demonstrated by the fact that the reaction efficiency was significantly reduced by the use of protein group without carbonyl group, such as benzene group. To extensive this reaction system, we next try the tripeptide synthesis. Specifically, we challenging the peptide elongation of dipeptide methyl ester, which was prepared by the above reaction condition. However, unfortunately, because the carbonyl group of protein group is far from the ester, the sufficient direct effect could not be obtained, and the yield of the tripeptide is only 41% yield. But from the extensive investigation, we could solve this problem by addition of the tetrapyryl ligand. The length from the carbonyl group to the ester is around 5 to 6 angstrom, which is similar between the, each metal catalyst coordinated by the bipyryl unit of this ligand. So it became clear that the addition of this ligand could locate each metal catalyst at the appropriate position. From this data, the tripeptide synthesis was carried out without the isolation of dipeptide methyl ester. Then a lot of amino acid could be applied for this reaction system. The high yield of the tripeptide was obtained in most cases. Furthermore, the tetrapeptide were also successfully synthesized by the use of the dipeptide ester instead of the amino acid ester in the final step. Okay, then having achieved the efficient use of protein group, we move on to the next target. As I mentioned above, the protein group is now the essential tool for selective peptide bond formation by suppressing the several side reactions, such as the reverse and self-condensation and the overreaction and cyclization. However, the natural amino acids are abundant in nature, so therefore they are inexpensive and readily available. But the amino acid with protein group at specific position need to be prepared by the radioselective protection reaction system, making them several dozen times more expensive. Therefore, there is strong demand to develop the selective peptide bond formation using the unprotected amino acid. Previously, Professor Shepard have already established the amidation reaction using unprotected amino acid, using the electron deficient boron reagent to form the 5-membrane intermediate with unprotected amino acid. However, maybe due to the difficulty of this reaction system, there are a few examples of peptide synthesis. From this background, we focus on the trimethyl aluminum and diimidazole silane, which are bearing the multiple leaving groups. These reagents have formed the 5-membrane intermediate smoothly, so we hypothesize that this intermediate could perform as an excellent coupling partner of peptide formation because they would fulfill the dual role of protection of amino group and the activation of C-terminal ester. As a result, the 5-member intermediate formed by the unprotected amino acid and each reagent could react with the amino acid tertiary butyl ester smoothly, especially for the aluminum one. The 5-member intermediate were added to the reaction solution, enabling the one-pot oligopeptide synthesis. Both of these systems have demonstrated the broad scope of substrate and have shown the value compared to the conventional method, at least the step of numbers. But there still are weak points, such as protecting group at side chain, where they protected in situ under the aluminum reaction condition. And these silent reagents need to be prepared in each time by distillation. So we next attempt to the improvement of this reaction condition. This diimidazole silane were prepared by the dichlorosilane and thiamidazole. So we plan to the one-pot reaction of diimidazole silane synthesis and peptide bond formation. As a result, the reaction leading to the dipeptide tertiary butyl ester turned out to be the minor reaction. But the major one is the silyl cyclic dipeptide synthesis. And this compound was coming from the decomposition of tertiary butyl ester of dipeptide by TMS chloride, which are generated during the diimidazole silane synthesis. I was so excited when this compound was detected, because this unique structural compound was a completely unknown product. And I strongly interested in the physical and chemical property and characteristics. After minor tuning of this reaction condition, the desired product was obtained in 91% yield. Using this reaction condition, we synthesized several silyl cycle dipeptide. From the investigation of many amino acids, including not only natural amino acid, but also the characteristic unnatural one, we could report over 50 examples of silyl cyclic dipeptide, and around half of them are exceeding the 19% yield. A part of them are shown in this slide. When we use the amino acid with tertiary butyl ester at side chain, interestingly, the decomposition of tertiary butyl ester is only occurred at the C-terminal, but remaining the ester at side chain completely. And the amino acid with secondary amino group, such as proline and N-methylalanine, also compatible with this reaction condition. Furthermore, the diaceroma is not observed, even if the phenylglycine were used as electrophile. But unfortunately, when we use phenylglycine, phenylalanine, tyrosine, and cysteine were used as nucleophile, a large amount of diaceroma was observed. However, this purity problem could be solved by the use of the cumulative ester, in which one of the methyl group were converted to the phenyl group. Okay, then this novel silyl cyclic dipeptide was synthesized by the sequential reaction of cross-condensation reaction and decomposition of tertiary butyl ester and cyclization. Then we next investigated whether the amino acid silyl ester formed in situ could be applied for this reaction system instead of the amino acid tertiary butyl ester, which is caused the generation of diaceroma. After evaluating the various silyl reagents, it was found that the combination of BSTFN and thiamidazole could effectively form the amino acid silyl ester. Then this reaction solution was added to the reaction solution of 5-member intermediate. The cross-condensation reaction at desired position proceeded smoothly, yielding the corresponding silyl cyclic dipeptide in 95% yield. Since the reverse condensation product and since the self-condensation product was not detected under this reaction condition, this is the first report of the selective peptide bond formation between the unprotected amino acid. using this reaction condition, a lot of amino acid could be applied for this reaction system, resulting in the isolation of over 80 examples of silyl cyclic dipeptide. And this compound was composed of the silyl N and silyl O bond, which are easily cleaved with the fluorine-based reagent. Therefore, these compounds are bearing the unprecedented potential to perform the peptide elongation at both ends. At first, we tried the C-terminal elongation with maintaining this fused ring structure. By starting with the thiamethimidazole, various amino acids ester could introduce to the diffused ring structure at high yield. Then, when these silyl cycle tripeptides were starting with the fluorine-based reagent and the electrophilic amino acid, resulting in the formation of the tetrapeptide by L-terminal elongation. The pentapeptide were also successfully synthesized by the tantalum-catalyzed C-terminal elongation before the N-terminal elongation. This pentapeptide synthesis system could be easily construct the pentagastrin and time benching sequence. And a simple depth protection system lead to the time benching in total 67% yield. Furthermore, the cross-condensation of these silyl cyclic tripeptides are also applicable for this reaction condition by the help of the tantalum catalysis. And finally, after the cross-condensation of silyl cyclic tripeptide, capped with the amino acid ester at C-terminal to afford the heptapeptide. And this synthesized heptapeptide will have common sequence of the semaglutide found in the semaglutide, liraglutide, exenatide, or other pharmaceuticals. By synthesizing this variable sequence, we believe that we could prove the usefulness of this structure. And this is a summary slide. We could demonstrate the amino acid protecting group directed remote peptide bond formation. Second, we found that the trimethyl aluminum and dimidazole silane could be used for the one-pot oligopeptide synthesis by using the unprotected amino acid. By controlling this reaction condition, we could establish the general procedure to synthesize the unreported silyl cyclic dipeptide. And this compound was also formed between the unprotected amino acid, revealing the new possibility for peptide synthesis. And this bioactive compound could be easily constructed by using the silyl cyclic tripeptide as a conversion reaction. Okay, this is the final slide, the very, very important slide. And this is these results are obtained through the communication with the laboratory member. So I thanks to the member, and especially for the Professor Hisashi Yamamoto, who is the mentor of this laboratory. Also, the financial support is essential for giving these results. So I thanks to them. Okay, then thank you very much for your kind attention.
Thank you very much, Professor Hattori, for the exciting talk.
Thank you very much.
Okay, let's move to the fourth speaker today. Next speaker, Professor Hyunwoo Kim from Korea, from the POSTECH, Pohong University of Science and Technology. Dr. Kim got PhD from the Korean Advanced Institute of Science and Technology, so-called KAIST, in 2018 under supervision of Professor Sugbok Chang. And then postdoc experience in Cornell University, and in 2020, Professor Kim joined the Edward Vermont University as an assistant professor in the Department of Chemistry and Nanoscience. Today, he's going to talk about electrochemistry unlocks new possibilities in unsaturated CC bond functionalization. Dr.
Okay,
Kim, start when you're ready.
Yep, ready. Thank you for really kind introduction, Professor Kobayashi. And as introduced, I'm an organic chemist by training and using electrochemistry to develop a new synthetic methodology. And I'm very happy to share one of my recent research program using electrochemistry, especially on the functionalization of unsaturated CC bonds. And I also really appreciate for the Thieme Chemistry for arranging all of this great opportunity to share my research. So shown here are actually primary areas of interest of our group. So looks like 3 separate topics, but these are actually more about 3 different perspectives on how we view organic chemistry research. So the first part is going to be a mechanism, second part is target, third part is going to be a concept. So I'd like to introduce a little bit more about this. So first, we focus on the design of new reaction mechanisms within established paradigms. So for example, we're exploring how electrochemistry can be used to design radical polar crossover mechanisms in a more novel and efficient way. And secondly, we're interested in targeting new functional groups. In particular, we've been working on introducing perfluoroalkyl functional groups by combining them with pharmacologically relevant motifs, such as difluoromethyl group, which has drawn considerable attention in medicinal chemistry. And finally, we're pursuing what I would call a conceptual advance. So specifically, we're aiming to expand the reactivity landscape of organic electrosynthesis by merging it with classical photochemical reactions. So I'll go into more detail on this later on this talk. And for the first part of my research, designing a new mechanism within established paradigm, we chose to revisit the Mukayama hydration paradigm, which is already very widely used in total synthesis. And it's pretty well known that this cobalt hydride species could be inserted into substituted alkenes in Markov-Nikov regioselective fashion to generate this cobalt-3 alkyl species, which is in equilibrium with this cobalt-2 radical pair. And traditionally, this kind of radicals could easily be captured by radicalophiles such as triplet oxygen to do hydration in very efficient way. And more recently, this cobalt-3 alkyl species has been known to proceed further oxidation to form its higher valent cobalt alkyl analog, such as cobalt-4 alkyl species, so that this cobalt alkyl bond become more polar, so that nucleophile can be engaged in this hydrofunctionalization methodology. This is kind of very cool approach, but one thing is that the oxidation of this cobalt-3 to 4 is in many cases rate limiting because it should only be achieved by metallic disproportionation, because you only use the catalytic amount of cobalt in your reaction media, and in terms of diffusion limit, this kind of bimetallic disproportionation would be the rate determining step. And 2 talented students from our group just came up with the idea that we can just change this kind of rate limiting oxidation step by changing this oxidation method to electrochemical oxidation. So we can just simply dial in the reaction potential by using potential step to give a right oxidation potential to the system, so that we can generate this kind of cobalt-4 alkyl species and sometimes cobalt-2 carbocation kind of solvent caged ion pair to do either intermolecular reaction or intramolecular reactions. So by merging a bunch of mechanistic studies, we were able to propose this mechanism. So I'm not going into too much details in the interest of time. Anyways, I'll show our mechanistic hypothesis and mechanistic proposal on this type of paradigm. So these cobalt silane species are quite well known to be oxidized by even by chemical oxidation to generate cobalt-3 species. In the presence of silane, this can form a cobalt-3 hydride species, and this kind of MHAT process is also quite well known to generate this cobalt-3 alkyl species, as I mentioned in the previous slide. And our hypothesis was that we can oxidize these species by using an efficiently by electrochemistry to generate this higher valent cobalt alkyl species, which can be entrapped by even by a weak nucleophile such as phenols to generate this aryl alkyl ethers. And these aryl alkyl ethers are, especially for the sterically hindered alkyl aryl ethers, are very difficult to be achieved by synthesized by conventional Williamson ether synthesis. So we thought that this kind of mechanism would be really a nice way to get an access to this sterically hindered alkyl aryl ethers. And I'm not going to bore you with the optimization, but I need to say that we are able to observe radical homocoupling products such as compound 8 here when we use chemical oxidants instead of electrochemical oxidation. So it is because the oxidation rate of this cobalt-3 alkyl species is rate limiting. So by just putting chemical oxidant, you can do this in very rapid way. But when we use electrochemistry, we are able to get an exclusive chemoselectivity towards our desired reaction, our desired mechanism. So after we established the mechanism, we really wanted to move on to more challenging transformations, such as four-membered heterocycle synthesis. So the mechanism here shown here is pretty much the same, but we changed the substrate into quite abundant source starting material, such as allylic sulfonamides, substituting allylic sulfonamides to do intramolecular hydroamination to form azididines, which is four-membered heterocycle, that nitrogen heterocycle. So we just put this substrate in our reaction system. We're able to generate the similar intermediate, which could be electrochemically oxidized to form its higher valent analog. And in this case, we know that cobalt hydride species can be inserted into alkenes in Markov-Nikov redox selectivity, so that we can kind of designate the position where carbocation is generated. So we can kind of dictate the position where we can do the cyclization, so that even kinetically challenging four-membered ring formation could be achieved by using our reaction mechanism. And here I want to emphasize that when we use homoallylic sulfonamides, which contains terminal alkene, gave the same product from the case when we use allylic sulfonamide, which is because this cobalt hydride species could be inserted in Markov-Nikov selectivity, so that more substituted cobalt will be inserted into more substituted carbon. So this is quite an interesting point of this work. And we could also do some cation probe experiment because we are kind of still we're on debate of which species will be the real kind of resting state in this reaction, because it actually depends on the substitution pattern of your substrate, because we have highly substituted alkenes in this system, so that this might prefer the formation of this free carbocation and cobalt-2 kind of solvent-caged ion pair. So we try to check the possibility of generating carbocation here, so that we put cation probe substrate under our standard reaction conditions. We did observe the formation of carbocation rearrangement products, and we also did deuterium scrambling test, and we're able to see that not more than 100% of deuterium was incorporated in the beta position, which means this MHAT process is irreversible. And also Faraday efficiency was measured, and it was found to be about 85%, which is moderately high. So what we believe is the oxidation efficiency of this system is pretty good. So I think this kind of supports electrochemistry is an ideal source of oxidant for this system. And let's switch the gear and talk about our second topic, developing a new target. And we are particularly interested in developing a new functional group with more than one diphomethyl groups. And the reason why we're interested in diphomethyl group is that, well, unlike a trifluoromethyl group, which is already very popular in pharma industry, the CF2H group has one extra CH bond in this like methyl-like structure, and the CH bond is very polarized, so that this can be included in hydrogen bond interaction. So this can serve as a lipophilic hydrogen bond donor, because the lipophilicity could be also controlled by attached fluorine atoms, so that this can serve as a bioisoster of methyl or other hydrogen bond donors. So this is getting more and more popular in pharma industry. And the reason why we try to develop a functional group possessing more than one difluoromethyl group in adjacent position was that there are already many pharma related compounds which contains vicinal hydrogen bond donors, but there were actually no general access to get this 1,2 bis difluoromethyl group containing functionality. So these are kind of mechanistic overview of our strategy. And so actually the key to success was to use this zinc difluoromethyl DMPU source as both anion and radical source. Actually, I didn't see this coming, but maybe she did. Well, we put this conventionally utilized transmetallating reagent, which could be used in Negishi type cross coupling. She just put this reagent into our electrolysis conditions, and we were able to see that this radical was generated upon electrolysis. So this radical is, once this radical is generated, this can react with alkene to generate a secondary alkyl species, which could be further captured by copper species. And this copper species can do a transmetallation with this zinc source to generate this kind of copper-3, higher valent copper-3 difluoromethyl analog, which are kind of well known to do the reductive elimination to deliver the second part of difluoromethyl group. And also trying to merge a bunch of mechanistic study, we were able to propose this mechanistic scheme. So this actually has a dichotomous role, like a dual role. So this could be oxidized on the anode to generate this CF2H radical, which can react with alkene to generate the secondary alkyl radical intermediate, and in which this radical could be captured by copper species to generate copper-3 alkyl species. And this can do a reductive elimination to deliver the second part of difluoromethyl group. And we did NMR study that this copper-1 analog could also do the transmetallation to generate this copper-1. And in the cyclic voltammogram, we were able to also assign the peak for the copper-1 to copper-2 oxidation as well. So this kind of constitutes copper catalytic cycle for this vicinal bis difluoromethylation reaction. And shown here are the scope, and my favorite examples are here in the bottom. And we were able to synthesize vicinal difluoromethyl analog of pre-existing drug molecules. So so far we didn't actually check the bioassay of these compounds, but we kind of hope in the future we can actually really expand the 3D space of compounds which contains vicinal bis difluoromethyl groups. And here's the final topic of today's talk, and we're trying to revisit kind of classical photochemical reaction in organic electrosynthesis to expand the reactivity landscape. So I'm going to talk in more detail about the mechanism of newly developed reaction paradigm. We actually try to revisit a very well-established classical photochemical reaction, such as 2+2 cycle addition. So starting from quinone, you can just photoexcite this species into biradical. It's triplet states, which could be written as biradical, and this can do a 2+2 cycle addition to form another well-established photon-induced intermediate. And for some reason, we first hypothesized that we can oxidize this species into even more electrophilic species like this. And the reason why we propose that this might be able to oxidize is that maybe this species has a very high propensity of having an aromaticity on this ring, so that we can separate charge and radical character to the end of these molecules like this, so that we kind of hypothesize that we can engage with very weak nucleophiles like nitriles or naphthols to make reaction with this kind of more electrophilic species. And then we were able to synthesize many functionalized heterocycles by using this paradigm. And the mechanism here in the circle were actually developed during our mechanistic study. It was not from our initial hypothesis, but I'm going to talk more in detail in later slides. And in order to check our initial hypothesis that we can oxidize photon-induced intermediate, we did a very simple cyclic voltammogram experiment, and we were able to see that a new peak arises upon irradiation of light. So when we just mix this quinone and alkyne, we were only able to get the quinone redox couple, but upon irradiation of light, we were able to see a new peak arises. And this is, of course, because of the generation of photon-induced intermediates, and we can oxidize this species into its radical cation species. And after this, we were able to design a reaction by using simply using an acetonitrile as a solvent. We were able to design a 3-component reaction. So once these two compounds do the cycle addition upon photoirradiation, we proposed that we were able to generate this para-quinone methylide intermediate in situ, and which could be oxidized into its more electrophilic radical cation analog. And this radical cation analog can make a reaction with this nitrile to generate this kind of intermediate, and some aryl shift and elimination, we were able to form oxazole species. But during the mechanism study, we were able to see that our reaction has actually more than 100% of Faraday efficiency. And we did some DFT calculation by collaboration with one of our colleagues. And we were able to kind of propose that this species itself has its own triplet states, which can engage in an electron transfer of an intermediate which is produced during the reaction. So we'll also be able to assign all the photophysics of this intermediate, compound number 3 here, and we were able to also check the lifetime of this intermediate, 12 nanosecond, which is long enough to do bimolecular electron transfer. And in order to do the bimolecular electron transfer, the persistence of this intermediate 10 should also be checked, and we were able to just simply check the persistence of this intermediate by checking a cyclic voltammogram of this compound, our product compound, because when we put some base to this molecule, and if you do the cyclic voltammogram, if that shows pretty good reversibility, we can say that this species is quite persistent, and we're able to see a beautiful reversible peak. So we were able to propose that this kind of redox chain mechanism is present in our newly developed mechanistic paradigm. And shown here are the representative scopes of our reaction. Although we have 3 different standard optimized reaction conditions, we were able to get the product, desired product, which can also be compatible with late-stage functionalization. And by using different type of nucleophiles, we were able to synthesize a different type of functionalized heterocycles as well. And our final target is to use some chiral phosphoric acid or chiral any sort of chiral acid to do a steric differentiation of this nucleophilic attack to this highly electrophilic intermediate, so that if we generate a chiral center here, put this 1-2, excuse me, this 1-2 aryl shift will go through an inner sphere reaction pathway. So we kind of expecting that there should be a preserved chiral information from these intermediates, so that we can generate some atropisomers in the presence of chiral phosphoric acids. So to make a long story short, we're developing a synthetic methodology from 3 different perspectives, and I hope I convince you that electrochemistry offers a uniquely powerful platform for organic reaction development. And I would like to thank all of our students. I couldn't have done this without our students and the funding source. And again, I would like to appreciate Thieme Chemistry, Thieme Journals to giving this really wonderful chance to share my research. Thank you very much.
Thank you very much, Professor Kim, for his very interesting talk. Okay, please close the session and move to the next speaker. Next speaker, Professor Xiaohui Yang from Beijing Institute of Technology. Professor Yang in 2015 from Nankai University under the supervision of Professor Kyurin Zhou. And then she went to United States as a postdoctoral fellow at UC Irvine, with working with Professor B. Dong from 16 to 20. In late 20, she returned to China and joined the faculty member of Beijing Institute of Technology. And 2024, she promoted to a full professor there. Today, she's going to talk about catalytic hydrofunctionalization of unsaturated hydrocarbons. Professor Yang, please start when you're ready.
Okay, thank you. Can you hear me?
Yeah.
Okay, cool. Thank you very much for your very kind introduction, Professor Kobayashi. Thanks for the organizers. It's really my great honor to have such a wonderful opportunity to show some of our research. So today I would like to share some of our research is about all the hydrofunctionalization of unsaturated hydrocarbons. Actually, most of the part is about the hydrofunctionalization of alkenes. So for the hydrofunctionalization of alkenes, it's basically adding a hydrogen and a functional group across to a double bond, so that you could synthesize a lot of very high value products. So this reaction has a lot of advantages, such as you could use very feedstock start materials, and the reaction is also very high atomic economy and also very high efficiency. So actually, a lot of chemists has pay attention to these reactions, and the different hydrofunctionalization reaction has been achieved. So however, right now there is still some challenges or limitations. For example, so the reaction type is limited. So most of them is focused on like hydroarylation or hydroborylation, hydrocellulation, as well as hydroamination. So actually for hydrofunctionalization with like oxygen group or halogen, it's very difficult. So the other things is control the selectivity is also difficult because you have to like the region control is difficult, as well as the natural selectivity control is also hard. So the third challenge is the product structure is usually monometers. So you can only get like one product. So that the hydrofunctionalization is continues to be a significant challenge. So when we start our group like 4 years ago, so one thing we're really interested in is about the hydrofunctionalization. So we were thinking about like there's two strategies. So one is could develop some new catalysis. So we could realize or achieve the direct hydrofunctionalizations. So the other thing is we also think about the shuttle catalysis. So Morandi has did a lot of great job by using shuttle catalysis. So basically this is like you could transfer the hydrogen and the functional group from the very easy start materials to the alkenes. So you could realize the transfer hydrofunctionalization. So these strategies you could avoid the use of highly toxic, expensive, or difficult to handle reagents. So with these two strategies, we actually achieve a few hydrofunctionalizations, including like hydrocarboxylation, hydrosalination, hydroamination, hydrochlorination, as well as hydrothiolylations. So today I'm going to share 3 stories about the hydrofunctionalization with like oxygen group, like hydrocarboxylation, hydrosalination, as well as the transfer hydrothiolations. So the first story I would like to share is about the asymmetric hydrocarboxylation. So one thing we're really interested in is to synthesize this, allylic ethers. So before it's usually used like a lean or alkyl with like very expensive metals such as rhodium or ruthenium with alcohol to achieve the hydrocarboxylation. So we were thinking if we could use like feedstock dyeing and by using the earth abundant metal catalysis to achieve this hydrocarboxylation. So actually there is some group already did this reaction but right now it's only can synthesize like a racemic product. So the challenge is because this allylic ethers, the cell bond is reversible. So it's very easy to activate by the nickel. And also the selectivity issue is also a challenge because there is a 2 double bond. So control the radial selectivity is also, a hard process. So with this challenge in mind, so my first student, he started his these reactions by using the nickel. So very luckily, we figure out when we're using this 2-phos ligand as well as solvent-free conditions, we could got very high initial selectivity as well as the radial selectivity. So from this figure, you could see, when we didn't use any solvent, the initial selectivities could be like very good and didn't decrease with time going. So however, when we start use some solvent like THF, so at the beginning, we also can got high initial selectivity. So however, with time going, you could see the initial selectivity is gonna go decreased. So this is because it's reversible. So during this process, we figure out the reversibility is inhibited under solvent-free conditions. So here is the sum of the scope. Actually, this reaction gives pretty good substitute scope. Here is I just chose a few. So different functional group could be tolerated. And here you could see a lot of like halogen or silane oil could be tolerated during this process. So as we also did a very detailed like mechanism studies, including kinetic study, NMR study, as well as the deuterium labeling experiments. So here, due to the time limitation, I'm just gonna give the reaction like mechanisms. So the first, like both the alcohol and diene gonna coordination to the rhodium to forming this intermediate. Then it undergoes ligand-ligand hydrogen transfer to forming this resting state, which is allylic nickel intermediate. Then it goes outsphere attack to give the desired product and go back to the nickel catalyst. So the reason why we think it's outsphere attack is not inner sphere is we based on the calculation, we figure out this process within outsphere attack is show a lower energy barriers. So the second story I'm gonna share is about hydrothioylation. So actually, thiol, as we all know, it smells very bad. And also because it's very, easy binding to the catalyst. So it's usually deactivated the catalyst. And also the thiol itself is unstable. So with time going, it's very easy to become the dimer. And also because the thiol is a little bit acid. So there is background reactions with the alcohol, with the alkenes. So right now, for the hydrothioylations, you could only achieve the very active alkenes. So compared to thiols, the thiol ethers is more compatible with the catalyst, and also it's stable. It's not easier, to decompose. Also, the smells is better, and also there's no background reactions. So we were thinking if we could use thiol ethers as the starting materials, and if metal catalysts could activate this carbon-sulfur bond, and then undergoes beta hydride elimination, if it can give this intermediate. If the alkene could exchange with this alkene and then undergoes insertion and reductive elimination, so if we could got the product. So this means before we were thinking, can we achieve the transfer hydrothioylations? So with this like hypothesis in mind, so one of my students, Zheng Wang, has started his research by using rhodium. Actually, he tries a lot of metal catalysts. So eventually, we figure out very interestingly, the rhodium could catalyze this process to give the transfer hydrothioylation product 3AA. So very surprisingly, we figure out, when we change the rhodium from the rhodium triplet to a cationic rhodium, like if we use like rhodium BF4, it gives a carbon thiolation product. Basically, it's the carbon-sulfur bond break and then adding to the double bond. So this we call it the carbon thiolation product. So this means the counter-ionize completely switch the chemical activity. So if we use a more like coordinate counter-ionize, it's give like the transfer hydrothiolations. So otherwise, if we use a cationic rhodium, it gives the carbon thiolation product. So first, of course, we also did the scope. Like for the carbon thiolations, the scope is kind of limited. So usually for alkenes, it's limited to the styrene. And however, for the transfer hydrothiolations, the scope is pretty wide. So both the like active alkenes as well as like this alkyl substituent alkenes and works pretty good. And for the thiol part, the aryl also the like kind of the alkyl thios all give very good results. So we also very like interesting why the cationize switch the selectivities. So actually we did a lot of mechanism study. So here I'm just gonna give the like over conclusion. So first, the rhodium, both two type of rhodium gonna like promote the cis bond like heterolytic cleavage. So we will got like a cation and also we can got a rhodium sulfur. So when we use like a triplet, because triplet can bind into the rhodium, so it makes the sulfur like more basicity, like the basic more basic. So the basicity is a strong basicity. So it could like promote the deprotonation, and it's in situ form these thiols which is binding to the rhodium, and then react with the alkenes. It can give the hydrothiolation product. So however, when we change like the cationic rhodium, because like the BF4 is didn't bind into the rhodium, so this sulfur is like the basic is not that strong. So it's the cation will react with this alkenes to undergoes like nucleophilic attack and give this intermediate, and then react with the rhodium sulfur to give the carbon thiolation product. So actually, we also did a lot of the calculation with Professor Shu from SIOC. So you could see when we use triplet, the sulfur is much more like, and the NPR charge is much more bigger than like this BF4 rhodium. So this kind of also like more prove our hypothesis. So the third story I would like to share is about asymmetric hydrosalination. So compared to thiol, salinose is more acidic, and also the BDE is smaller. So we were thinking if we could use metal to activate this salino hydrogen bond, like form the metal hydrogen. If we can form this intermediate, then it could reactive with this alkene, undergoes like insertion, and then undergoes reductive elimination. If we could got like this asymmetric hydrosalination. So we want use like its relatively strong acidity and lower BDE to forming this metal hydride. So very likely we figure out rhodium can also do this process. But right now, the styrene, the alkenes is limited to styrene, and the salinol is only, aryl salinol could works pretty good. So it can give good result about the hydrosalination. So when we do the mechanism studies, we figure out we could see the rhodium hydride in the proton NMR. You could see it very clearly. Even we could see in the room temperature. So based on this, and also over like KIE and also the kinetic study, we figure out the first step should be oxidative addition to form the rhodium hydride. And then the alkene binding to the rhodium and undergoes insertion to give this intermediate, which is the turnover limiting step based on the KIE. And at last, it undergoes reductive elimination to give the desired product. So this is like the 3 stories I would like to share. So basically, is we want to develop the different catalyses to achieve virus hydrofunctionalization, so that we could construction of virus carbon hydroatom bonds. So with that, I would like to first, of course, thank my student. So today I share 3 stories is about Qi, Zheng, and Yixin's work. And also I would like to acknowledge, thanks my supervisors. They always, very support to me, and of course the fundings as well. So this is our university, and thanks very much for your kind attention. And also welcome to China, and also welcome to our university. It's Beijing Institute of Technology. Thank you so much.
Thank you very much, Professor Yang, for the exciting talk. Okay, please close the slide. Thank you. Okay, let's move to the final speaker today. Final speaker is Dr. Durga Prasad Hari from Indian Institute of Science, Bangalore. Dr. Hari received his master's degree from Indian Institute of Technology, Madras in 2010. He then moved to the University of Regensburg, Germany, where he pursued his PhD with Professor Burkhard Koenig. After taking PhD, he joined Professor Jerome Basser's group at EPFL, Switzerland, as a postdoc, and then second postdoc to the United Kingdom with Professor Belinda Agabor, University of Bristol. Since 2021, he holds an assistant professor position at the Indian Institute of Science, Bangalore. Today, he's going to talk about carbon mimics from strange links. Please start when you're ready.
Thank you, Professor Kobayashi, for the kind introduction. Can you hear me?
Yeah.
Okay. Hello, everyone. Welcome to my presentation. Today, I'm going to talk about carbon mimics from strain rings. Before going to talk about this particular topic, I would like to give you a brief overview. The research teams that we are working at IISc Bangalore. So we are particularly interested in working on the carbon chemistry, especially development of selective generation of either singlet carbon or a triplet carbon, and controlling their subsequent reactions. And we're also interested in developing the strain-enabled reaction discovery, especially harnessing the high reactivity of strain molecules to access medicinally relevant compounds at ambient conditions. The tools that we use for driving these transformations are mainly light and electricity. But when we need these compounds with a high stereoselectivity, we are also interested in developing the metal catalyzed reactions. So today, I'm going to show our recent contribution to the strain-enabled reaction discovery using transition metal catalysis, especially generation of the carbons from these strain molecules. Why carbons? And most of you know that the carbons are versatile reactive intermediates in organic synthesis, and they participate in variety of rearrangement. The well-known one is the Wolff rearrangement, where we access the highly reactive TT intermediates. And they also participate in the XY insertion reactions to create the molecular complexity. And when you have a olefin or alkyne, they undergo 2+1 cycle addition to give the cyclopropane or a cyclopropene. And they also form the ylide intermediates, which can further participate in multi-component reactions to generate the molecular complexity in no time. The one question that we ask is that how we generate these carbon intermediates. When you look into the literature, most of the time, the people use a diazo compounds with either metal or a light where you can access these carbon intermediates. And you can also use a triozones using a metals where you can also get the carbon intermediates. The another commonly used carbon precursor is a sulfoxone halides. And when we start our research group 4 years ago, we asked one question that can we generate the carbenes from a strain rings? So why we are interested in the generation of carbenes using strain rings? Because the carbon chemistry is very well known and a lot of transformations are reported. If you can come up with a new carbon precursors, you can visit the carbon chemistry, which will open up a new chemical space. So with that, so we in the last 4 years, we are able to find that the strain rings like a bicyclic butane or a propylene, they're able to give the carbon intermediates when you react with the metal complexes. So today, I'm going to tell you 3 stories. In the first story, I will tell you how we use the metal carbon chemistry to harness the carbon reactivity of a bicyclic butane to access the skip dienes in a highly stereoselective fashion. In the second story, I will talk about how we generate the carbenes from a propylene and develop the first sigmatropic rearrangement to access this allylated methylene cyclobutanes. And then I will finish my presentation by showing you how we use the propylene carbene chemistry for editing the cyclic ketones. So let me start with the first story where we harness the carbon reactivity by cyclobutane. So the bicyclobutane is among the strain molecule and which has 66 kcal per mole. And the bicyclobutane chemistry is very well established, especially where the reactions can take place at the bridgehead bond. And difunctionalization is something that is very well known, where you add a nucleophile and followed by trapping the anion with an electrophile to difunctionalize this carbon-carbon bond. Or you can use a different radicals also to difunctionalize this bond. That leads to the synthesis of tetrasubstituted cyclobutane derivatives. So another common reactivity that also well explored is insertion of carbene into the strain bond to give you the bicyclopentanes, which are bioester of benzene ring. And they also recently, there are several reports that they also participate in 2+2 or 2+3 cycloaddition reactions to access a bicyclohexane or bicycloheptane derivatives. So here, we wanted to invoke the different reactivity of bicyclobutane, especially the carbon reactivity. Why? Because such kind of carbenes are very challenging to access using traditional methods. And the second is that such reactivity is underexplored. So what is our hypothesis here? Is that starting from this bicyclobutane, so we wanted to use the metal carbon chemistry to invoke the carbon reactivity to access this kind of a skip dienes. Let me give you the mechanism in more detail. So we start with the metal. So when the metal reacts with the diazo compound, you form a metal carbenes. The metal carbenes are usually highly electrophilic. Now we have this bicyclobutane, the strain bond. Here, the strain bond acts as a nucleophile and it adds onto the carbon center of electrophilic metal carbene and give you this kind of a Twitter ion, which is very unstable. And also due to this puckered structure and very rigidity, and we also expect that it will majorly give one stereoisomer here while regenerating the metal catalyst. So if you look at this bicyclobutane, it's overall, it is acting as this kind of a formal carbene. And now this carbene cross dimer is the metal carbene and to give you this kind of a skip dienes. Why skip dienes? The skip dienes are present in various natural products. And also they are very important building blocks in the total synthesis of complex natural products. So with that, we started this project. I'm not going to talk about the optimization details, but what is very important here is that we need a rhodium catalyst in DCM at room temperature. We got the desired product in good yields with excellent stereoselectivities. The scope of the reaction is very broad. So you can use a variety of diazo compounds, especially donor acceptor diazo compounds. And also diazo compounds having a complex substrates also worked. This chemistry is not limited to bicyclobutane having an ester. You can also extend it to bicyclobutane having a ketones or an amides. In all cases, we obtained good to excellent yields and always we observe a single diisomer. So that brings me to my second part, where I'm going to show you how we generate the metal carbene from a propylene and for the 2,3 sigma tropic rearrangement. The propylene is again another strained molecule and which has 100 kcal per mole, more strained than the bicyclic butane that I showed a few minutes ago. Again here, the strained bond, the middle bond is the bond which is more reactive, and there are a lot of literature reported on the breaking of this carbon-carbon bond to give this kind of a 1,3 difunctional product, which is a bioester of a benzene ring. So what we are interested in the carbene reactivity of propylene and to get access to this kind of a methylene cyclobutyl carbene intermediate. Again, we are interested for two reasons. The one is this kind of a new reactivity of the carbene reactivity of 1,1 propylene is less explored, and accessing such a carbene intermediates from a classical or traditional methods is highly challenging. So here is our hypothesis starting from this propylene. So we want to use a metal to activate a propylene to give a methylene cyclobutyl carbenes, and then use them in 2,3 sigma tropic rearrangement to access this kind of a functionalized allynyl methylene cyclobutanes. How does it work? So when the metal activates the propylene and it gives this kind of a metal carbenoid, and which is an electrophilic, and we have this propyl sulfide, the lone pair of sulfur will add to the carbon of the metal carbene to give you this kind of an halide that undergo 2,3 sigma tropic rearrangement to release the metal and give you this kind of alienated methylene cyclobutanes. Again, if you look at the propylene here, so this is kind of a equivalent of this methylene cyclobutyl carbene. So why methylene cyclobutanes? These methylene cyclobutanes also present in a variety of natural products, and due to the strain of this methylene cyclobutane bond, this also participate in variety of cycloaddition reactions, ring expansion to give the complex molecules. With that, we are interested in this project. Again, I'm not going to talk about the optimization due to the time constraints. So what we need here is that we need a copper catalyst here. And starting from the propyl sulfide, we are able to access these compounds. The scope of the reaction was quite robust. So we can use the propyl sulfide where the phenyl group is substituted on the sulfur. And this is my favorite example, where we start with the propyl sulfide having a bicyclopentane and reacting with this kind of a carbene, and you get access to this kind of a molecule where there are two strain rings are there. One is a methylene cyclobutane and another is a bicyclopentane. And we are also able to access this kind of a disubstituted allene when we have substitution at a propylic position. And this chemistry also extended to the selenium compounds. Next, we also tested Li sulfide in the same reaction. And also the scope of the reaction was very broad. You can use a variety of Li sulfide having a substitution on the sulfur, such as the heteroaromatic, and you can also have the cyclic ketones and the long alkyl chain having fluorinated substituents. And this is again another my favorite example, where we started from a bicyclic pentane having Li sulfide that reacts with the propylene to give you this compound where again you have these two beautiful rings in the same molecule. So that brings me to the last part of my presentation, where we use the propylene chemistry for editing cyclic ketones. I don't have to give introduction to ketones, and the ketones are very important building blocks, and there are solvents, and they're present in enriched drugs, and also we use them as a feedstocks. So when we want to edit or do the ring expansion of ketones, and in 1885, and it started that you can do an homologation with a diazo compound where you insert a carbon atom into the cyclic system. And later on, you can also insert a nitrogen atom into the cyclic ketone or acyclic ketone, which we call as a Beckmann rearrangement. And similarly, if you wanted to introduce an oxygen atom, this is none other than the Baeyer-Williger oxidation reactions. So these are very well known, and this is a textbook chemistry. What we wanted to do in 2025, can we use the propylene to edit the cyclic ketones where now you can access directly a spiro compounds having this methylene cyclobutane, and this methylene group can be further functionalized to have access different functional groups. So with that, so here is our hypothesis. Starting from this propylene, so we want to use a methyl to activate the propylene and along with the cyclic ketones to access this kind of a spiro compounds. So let me give you more detail about this mechanism. So we start with this Lewis acid, a methyl. So it coordinates to the oxygen and to increase the electrophilicity of the carbonyl of the carbon here. And now this carbon is electrophilic. Now you use a strain ring, propylene. It acts as a nucleophile, add onto the carbonyl here, followed by rearrangement to give you this kind of a methylene cyclobutyl carbocation. Now you can see that this intermediate can go for ring expansion and to give you this kind of a spiro compound. And you can see that propylene here act as a equivalent of carbene, where the negative charge here add to the carbonyl and give you this kind of a carbocation intermediate, which undergo ring expansion to give you this kind of a the spiro compound. So with that, so we optimize the reaction condition, and we found that scandium is the best catalyst for this kind of a ring expansion reaction. And the scope of the reaction is remarkable here. So we can use a variety of cyclic ketones. And here, one of the spiro compound we are able to synthesize in 56 grams, and this will be soon commercialized at ENMine Limited. And this is not limited to the simple small rings, and we can also use a 6-member ring containing oxygen. Again, we scaled up this reaction in 32 grams. And you can also use a larger rings, and you can also access these spiro compounds. Unlike in other skeletal editing, this is very special editing because you can do the iterative process. Starting from this 6-member cyclic compound, first we performed our editing reaction to introduce this methylene cyclobutane, and then we did a ozanolysis to introduce the carbonyl group here. At this stage, if you see this spiro compound, we have 2 cyclic ketones. One is a 4-member ring and another is a 7-member ring. But due to the ring strain, now when we use this propylene and scandium triplet, we're able to edit the 4-member ring, and it became a 5-member ring with a methylene cyclobutane here. Again, we repeated this ozanolysis to access the cyclobutanone, and now we have 3 rings, but still the cyclobutanone is more strained. This undergo for further ring expansion reaction. Now you give 7-5-5 spiro compound with 1:1 DR. We stopped it here because here we have 2 chiral center, and then it start giving the diastereomers. So that's the end of my presentation. In the last 15 minutes, what I showed you is that classically we use diazo compounds or triazoles or sulfoxonium halides for a carbene generation. What we showed here is that you can also use the strain rings for carbene generation. These carbenes are challenging to access using a traditional methods. And I showed you 3 different stories. In the first case, we use the metal carbene chemistry to harness the carbene reactivity of BCD to access skip dienes. And in the second story, I showed that how we generate the metal carbenes from propylene and use it in the 2,3 sigma tropic arrangement. In the third story, I showed how we use the propylene chemistry for editing the cyclic ketones. So with that, I would like to thank my research group and also funding agencies and Thieme once again for this opportunity to present our research work. And also, I would like to thank all of you for being here and your kind attention. Thank you.
Thank you very much, Dr. Hari, for the exciting carbon chemistry. Okay, I also like to thank all 6 speakers today for their excellent talks. And also thanks for joining the web seminar. Karen, do you have any comments in the closing?
Yes. Yes, I'd also like to say thank you to you, Shu, for chairing today. Thank you again to all of the 6 speakers for their insightful talks. I learned a lot. And remember, if you missed anything, you can go back and watch the recording later. And if you posted any questions, also go back to the Cassini page and follow the chat for answers. And finally, I'm going to put this in the chat as well. Our next seminar is only a couple of weeks away on the 10th of December. So if you're interested in small molecules in medicinal chemistry, please sign up for that as well. And I hope to see you there. Thank you all very much for joining us this morning. Thank you. Bye-bye.
Okay, thank you very much. Bye.
Thanks. Bye.